Hey everyone, I'm Alexander Miller, an undergraduate student researcher in the Bruce Bowerman Laboratory at the Institute of Molecular Biology. Now there, I've been screening for genes that are required for proper embryonic morphogenesis in the nematode Caenorhabditis elegans. Now morphogenesis is just the coordinated cell movement and the shape changes that occur during the development of an organism. And this process is conserved across all multicellular animals, which makes sense. If you have more than one cell, you want to be able to specify what shape those cells need to be in and where they're going to go in order to create the final structure of a tissue. Now, this is vitally important as we know that the structure of a tissue is integral to its proper functioning. Now, defects in morphogenesis can cause a wide variety of different pathologies, ranging from the inconvenient to the lethal. One example of this is spina bifida, which is a congenital birth defect causing the outpocket of the spinal cord during birth. And this is just due to the improper closure of the neural tube during development. Now, spina bifida is relatively easily cured or treated with surgery, but there's other morphogenesis disorders that are more extreme and also untreatable. So clearly it's important that we better understand morphogenesis to provide treatments for these sort of disorders. I'm looking at morphogenesis in C. elegans for a number of reasons. C. elegans is a great model organism to study your own biology. It's genetically similar to humans. Its life cycle is small and manageable. It's transparent, and it's also very easy to maintain. Now, using C. elegans, I've been screening for the genes that are required for this embryonic morphogenesis. And I'm doing this using a collection of temperature-sensitive mutants that we have in the Bowerman laboratory. Temperature-sensitive mutants are just mutants that only display the mutant phenotype when exposed to a certain restrictive temperature. And this allows us to bypass any prior gene requirements that would otherwise interfere with our studies. Now, using this collection of temperature-sensitive mutants, I've been running or working with a 4-genetic screen to identify the genes that are required for embryonic morphogenesis. Now, this is done first by selecting for those temperature-sensitive mutants that are morphogenesis defective and then genetically characterizing them to select for those that are recessive and single mutant. After this, we can identify the causative mutations by running a combination of whole genome sequencing and single nucleotide polymorphism mapping. And this is done to create a comprehensive list of genes that we know to be related to embryonic morphogenesis. Now, what we found so far is that the large majority of genes that we've identified are somehow related to gene expression regulation. Now, this is all done to further our own understanding of human development, as well as to figure out possible treatments for these morphogenesis disorders that I was talking about earlier.